Hello everyone, my name is Raúl Fernández and I am a researcher at Hospital de la Princesa in Madrid. Today I would like to introduce the topic of our poster. Bioinformatic approach firstly uncovers the relationship among primary cilia and PitNETs worst prognosis. PitNET is the abbreviation for pituitary neuroendocrine tumor, which are neoplasms that develop in the pituitary gland. They represent the second most frequent intracranial tumors. The location of these tumors within the pituitary gland is significant because it can lead to hormonal imbalances. Clinically, PitNETs can be classified into two major groups: functional PitNETs, which cause hormonal disorders due to the hypersecretion of one or more hormones, and non-functional PitNETs, which do not result in hormone hypersecretion but can cause common symptoms such as headaches or blood pressure. Our research group previously identified a novel cellular structure, the primary cilia, which has not been described in this type of tumor before. Primary cilia are a mechanosensitive antenna developed by epithelial cells. Now let's discuss the objectives of our work. 1. Explore and analyze the association between gene sets associated with ciliogenesis and primary cilia in these two subtypes of PitNETs. Second, investigate the most dysregulated genes involved in the ciliogenesis process that have relevance in PitNETs. Third, evaluate and select specific genes with biomarker characteristics to predict the prognostic evolution of PitNETs. And the last one, validate the selected genes expression and localization using immunofluorescence methods. To accomplish our research goals, we followed these steps. First, we collected public data from two repositories. The first one, the Gene Expression Omnibus, GEO, using microarray technology for gene selection related to ciliogenesis. And the second repository, the European Bioinformatic Institute, EBI, to validate the selected genes with RNA sequencing technology. The second step was immunofluorescence assays were performed to validate the selected biomarkers associated with the ciliogenesis progress. To that goal, we used a cohort of 118 patients recruited from Hospital de la Princesa, and tissue microarrays were utilized for these assays. Let's take a look at some of our key results. Our initial selection of 155 cilia-related genes revealed that patients with clinically distinct PitNETs and normal pituitary samples can be clustered separately based on their gene expression. Then we conducted a differential expression analysis across all comparisons and identified two dysregulated genes and with well-described function in the ciliogenesis process. The first one, double domain containing 2, also known as DCDC2, and the centriolar colloid coil protein 110, also known as CCP110. We further investigate the selected genes separately and discovered that DCDC2 and CCP110 exhibit an inverse pattern of behavior between two groups of PitNETs. This suggests the involvement of different regulatory mechanisms in the process of ciliogenesis. To validate the observed expression pattern, we utilized the RNA-seq repository, confirming that the expression pattern of both genes encompasses diverse types of PitNETs. Moving forward, we performed immunofluorescence assays to examine the selected gene, DCDC2. Our findings revealed its location at the tip of the primary cilia in functional PitNETs, whereas in non-functional PitNETs, its expression was observed through the entire cilia structure. To summarize, our work led the following conclusions. Expression patterns of genes related to the ciliogenesis process allows stratification of patients according to the tumor's endocrinology in PitNETs. Second, inverse relationships exist between the expression patterns of DCEC2 and CCP110 genes and the endocrinological classification of PitNETs. Third, diseases associated with functional PitNETs exhibit higher CCP110 expression and lower DCEC2 expression compared to the other subtypes. And the last conclusion, immunofluorescence validation indicates reduced expression of DCEC2 in functional PitNETs, while its expression extends throughout the entire cilia structure in non-functional PitNETs. Thank you for your attention.